Hello everyone, my name is Guilherme Tergino Valente. I am professor at São Paulo State University, UNESCO, and the head of SBGL, the System Biogenomics Lab, and I will present our most recent paper. This paper is called Predict Metabolic Pathway of Plant Enzymes without using sequence similarity models from machine learning. In this paper, we developed and present a new tool to classify the metabolic pathways of plant enzymes. But the good thing of this paper that we don't use sequence similarity by different reasons, and also we predict the metabolic pathway and not the enzymatic reaction number that is the focus of most of papers. And then this paper is also an open access paper. You can find the paper here, and the tool is published here in this link. So let's go to see what we did. Well, as probably you guys know, after a sequence a genome, we pass by a process of genome annotation, and also we have a draft of the genome or the genome as a whole. But the problem is most of process to annotate genomes are based on sequence similarity approaches, and then but we can have many problems with this process because we have many species specific subunit functionalization and lack of motif domains in many different genes and proteins, and then we can have a bunch of annotation flaws in the genome because despite the problems of the species specifics, for example, the approach based on sequence similarity is not useful for many different genes or proteins, right? Then the functional annotation of proteins is quite not so easy. So in summary, annotate genome is still a challenge task. And enzymes comprise a huge part of the proteins of a genome, of a species, ranging from 15 to 35% of the proteins. It works in most of metabolic reactions, right? As we can see here in CAG pathway, for example. And then the interesting thing concerning enzymes that they do specific reactions, precise reactions. And enzymes, if we know the function of enzyme, we can do metabolic engineering. That's really, really important and interesting for the society as a whole. But functional annotation of enzymes are still hard by the reasons that I already mentioned. And well, here I can do a kind of interesting idea, like what I say, the life equation. Here's a complete metabolic pathway, right? Metabolic map, I mean. And what is a metabolic pathway? Is a subset of the whole metabolic system. And metabolic pathways harbor different enzymes and different reactions. And interestingly, EC number is a set of 4 numbers are used to depict the reactions of enzymes. But to assign reactions doesn't mean that you have the idea of the metabolic pathway. They are not synonymous. In fact, different enzymes can have the same EC number, and different metabolic pathways can have the same reactions. So it's one thing that we need to be very clear in our mind. EC numbers are not metabolic pathways. So EC numbers to assign EC numbers for proteins are the focus of most of bioinformatic tools concerning functional annotation of enzymes. But mApLe, the tool developed by us and presented in this paper, it does a metabolic pathway prediction and not assign EC number, and interestingly, without use sequence similarity, make mApLe able to predict metabolic pathway for a huge set of enzymes. So, what mApLe is beyond prediction of metabolic pathway? It's a machine learning based tool, do not use sequence similarity. It has a high predictive performance. It's not biased by genome side, neither species. Classify sequence without EC number as well. Classify sequence without homologs, and we claim mApLe as a complementary tool for annotation, the genome annotation. We don't say that the mApLe can substitute most of tools, of course not, but it's a complementary tool that can give you additional information. Just a general view of how we developed the mApLe. The first thing that we did is collect sequence from the UniProt database from different species, plant species. And then we reduced the redundancy of sequence. We excluded sequence with poor annotation, and we excluded the pathways that had a few amount of proteins representing this pathway. Because in first assays we saw that we must have a minimum number of around 50 enzymes of a single pathway to have a reasonable predictive model. And then after this, we calculated physical chemical features. Well, as I said, we are not using sequence similarity. Then we calculate physical chemical features to have a matrix to be an input of the machine learning, right? And then we performed attribute selection to reduce the universe of attributes. And then we also used different algorithms like support vector machine, neural networks, trees to make the training and create models. In the figure 1 of the paper, you'll see the general pipeline used to create the mApLe. So after we have the set of instance, the sequence, right? We split in two different sets. One of them, we create the training set, test set, and evaluator set. And another one, we created the mApLe test set. The training set was used to create the models per se, and the test set, evaluator set, and mApLe test set were used to evaluate the performance of these models and to make more in-depth adjustments. After to select the algorithms, we combined the models. We combined the algorithms, the most important attributes, features, physical chemical features. We did a hyperparameters adjustment. We combined algorithms, as I said. We created 15 models at the end of this whole process. And then we always tested these models and adjustments, so on, in the training test, in the test and evaluator and mApLe test data sets to have a way to do a fine tuning of the parameters to have the best model and to avoid overfitting, bias, so on, right? In the paper we'll find some tables and figures, but I will highlight some tables, the function of some tables. In the table 1 of the paper, you will find let me get me out. Yeah, you will find the function of each data set that we built, the number of enzymes, and the number of species that are part of these data sets. In the table 2, you will find the metabolic pathways choose in the end of the whole process, the number of instances positive instance, the number of data sets, so on. In the table 5, we will find the metabolic pathways and the predictive performance. We can see an average around 87%, excluding the 3 metabolic pathways that were not so good, need some improvement. And then at the end, we have 12 metabolic pathways with an average of correct classifying instance around 87%. And in the figure 4, we will find the predictive performance of these uh, pathways, these models over test and evaluator sets, right? Indeed, we present here classification of instances that are unknown for the models. We can see a very high predictive performance, right? And with this data, we also confirmed that the mApLe is not based, is not has not doesn't have a bias concerning species or some peculiarities of species or homologies, so on. In the supplementary figure C, you can see details of lots of different statistical parameters that we use to evaluate the performance of mApLe. Then I advise you to go in the supplementary materials, mainly supplementary tables that we present lots of details of our evaluation. Okay, that make our paper reliable, describing a reliable too. Put me again. Where's me? Oh, yeah, here. Yeah. Okay. So finally, where can I find mApLe? It may be a question. Surprise! You guys can find mApLe as a user-friendly interface. In this case, you can just click in this link here and you can download the mApLe. You will find this page here with a manual, a tutorial how to install and execute the mApLe. You will find example, you'll find lots of different information here. Here in this part of the figure of the page, we depict the part of the mApLe output. The mApLe, after you execute the mApLe, you will have the predictive performance over your input, but also a spreadsheet of gold standard enzymes, the classification of over gold standard enzymes. And you can use this spreadsheet to evaluate your data, your results, and see how good was the performance to classify your set. It's very interesting because you can have a kind of way to evaluate the performance of your data, right? So mApLe can use as input a fast tab or CSV file and output a spreadsheet that you can upload in Excel, for example. Here's the details of the spreadsheet, as I said, and in the end of the manuscript, in the last paragraph of the manuscript, you'll find guidelines how to filter your data and make your own model. If you're interested to build your own model, you can do it. You can do it using the guidelines that we present in the paper, right? So this is the mApLe. Hopefully you will enjoy this paper and this algorithm, this program. And if you want to join us, to join the System Biology Genomic Lab, just contact me, okay? Cheers!